कहाँ तक कराया था? Come front, no? You can go pay. Yes. So in the last class, we were discussing about the molecular basis of inheritance, and in this chapter, we have studied the two types of nucleic acid. One is deoxyribonucleic acid, and one is ribonucleic acid. Unable to listen. Yes. So in the last class, we were discussing about the molecular basis of inheritance. And in this chapter, we have studied two very important experiments. One is the transforming principle or Griffith experiment, and second is the Hershey's and Chase experiment. So after studying these two experiments, the confirmation was done that DNA is the genetic material. And already we have studied the chemical composition of DNA. So today we will discuss DNA replication. To study DNA replication, we need to recall what we have studied about the double helix structure of DNA. Yes. So this double helix structure of the DNA was proposed by two scientists. Can you name them? Yes. Yes. Watson and Watson and Crick. Yes. So these two persons have proposed the structure of DNA as double helix structure of DNA. According to them, what they have done, they have taken the X-ray diffraction data from two scientists, that is Wilklin and Franklin. So in 1953, they have proposed the double helix structure of DNA. And according to this model, to understand DNA replication, we should know the structure of DNA. So what they have proposed, they have clearly defined the structure of DNA. Yes. Point number 1, 5 dash, 3 dash, 3 dash, 5 dash. According to the Watson and Crick model, point number 1, the double helix structure of the DNA consists of two polynucleotide strand. It consists of two polynucleotide strand. The backbone of the DNA is made up of sugar and phosphate. The backbone of the DNA helix is made up of sugar and phosphate, and in between we are able to see nitrogenous bases. We are able to see nitrogenous bases. So these are nitrogenous bases. So here, when we study the DNA helix, so usually at least one DNA will be of 10 helix. It is on an average made up of 10 helix. And if you see the, if you take one helix, okay, if you take this one helix, no, in this one helix, usually 10 base pairing is seen. How many? 10 base pairing will be seen. And if you measure the distance between the one base pair and another base pair, it is 0.34 nanometer. Is it clear? It is 0. Nanometer. And if you take totally 10, no, the size of the helix will be approximately 3.4 nanometer, or we can also tell 34 Armstrong. 34 Armstrong. And like if you take the diameter, so here we have adenine, here we have thymine, here we have guanine, and here we have cytosine. If you measure the distance between these two polynucleotide strands, no, it will be 20 Armstrong unit, that is 2 nanometers. It will be 2 nanometers. Maximum DNA will be right-handed coil structure. The helix will be in the right-handed form. Okay. In few, right and left both are seen together. Okay. If the complete DNA, whether it is 5 helix or 10 helix, how many helix are there? What is the length? It can be a short DNA fragment also, long DNA fragment also. If all are right-handed, no, that time we will call it as BDNA. It is called BDNA. If one helix is showing right direction, one is showing left direction, right, left, that time such DNA will be called as ZDNA. It will be called as ZDNA. Is that clear? And so here two polarities will be seen in the DNA helix. One will be 5 dash to 3 dash, 5 prime, 3 prime we will tell. Okay? Polarity, and one will be 3 prime to 5 prime polarity. Done? And the DNA helix will maintain one rule called Chargaff rule. According to the Chargaff rule, no, in the DNA helix, always the one purine will base pair with one pyrimidine. One purine will base pair with one pyrimidine. So here, if you take adenine, one purine, it will base pair with thymine. Okay? And here that will be bonded by hydrogen bond. It will be bonded by hydrogen bond. Is that clear? It will be bonded by hydrogen bond. And similarly, if you take guanine, it will be base pair with the cytosine. So here adenine and thymine are bonded by two hydrogen bonds, two weak hydrogen bonds, and guanine and cytosine will be bonded by three weak hydrogen bonds. Is that clear? If you take one polynucleotide strand bonding with another, inside base pairing is done with the help of a bond, that is hydrogen. No, if one polynucleotide strand is paired with another polynucleotide strand, no, from outside, this is one strand, this is one strand. Nay. So here we have one phosphate, here we have one phosphate. Now this phosphate also will be bonded. Is that clear? So when that pairing will be done, it will be bonded by phosphodiester bond. When two base pairs are bonded, they are bonded by hydrogen bond. Okay. When two polynucleotides are bonded, it will be bonded by phosphodiester bond. Is that clear? So here, these are the few characteristic features of the double helix model. Okay. So now if you know this, we can go with the replication process. Any doubts you have? Yes? No doubts. Shall we proceed with the DNA replication now? Yes? Now, so coming to DNA replication. We know the structure of DNA now. What is this replication means? Yes? What do you understand by the word replication? Yes? Copying? Copying? Yes? Copying? Producing its own replica? Yes? Producing its own replica? So here, what exactly happens in DNA replication? So from one parental DNA to daughter DNA will be synthesized. Is it clear? From one. So here, DNA replication can be called as producing its own replica. Producing its, we are taking one parental DNA. From that parental DNA, two newly synthesized DNAs will be produced. So how this will happen, sir? So if you see how the replication process start, assume this is the DNA helix. Yes, this is the DNA helix. No? Now, for the replication process, when this replication process take place in DNA, any idea? Yes, you have studied in first year PUC also. Cell cycle and cell division chapter. No? No idea? In cell cycle and cell division, you have studied two phases. Do you remember? Yes, that also don't know. Huh? Two types of cell division you have studied. Yes, mitosis and meiosis. Okay. Go for mitosis. Okay. Once the mitosis is over, which process will take place? Yes, directly Jeevan. What is the phase called? Yes, interphase. Interphase. The cell is preparing for cell division. Nay, once the cell is ready, it will undergo mitosis. After mitosis, again the new cell will be preparing for cell division. Nay, in the interphase, phases will be classified. G1, S phase, G2 phase, G0 phase. Okay. If the cell doesn't want to undergo cell division, it will enter into G0 phase. Okay. So here in the interphase, in the S phase, during the interphase, in the interphase, in the S, that is synthesis phase, we will tell. Is it clear? In that phase, the DNA replication will take place. Is it clear? So here, copying the DNA, making its own replica. So for that DNA replication, when the DNA replication start, first what exactly happens? Yes, what will happen? Yes, breaking of hydrogen bonds should take place. Yes, breaking of hydrogen bonds should take place. So once the hydrogen bonds are break down, it will become like this. Yes, it will form like this. Yes. Now, so to undergo with the replication process, there are few enzymes needed. Breaking of hydrogen bond will not take place by its own. It will take place by its own. No. Right? Somebody has to do that. So enzymes, list of enzymes are needed. Number one, first we will tell unwinding of DNA helix. The helix which is present like this, no, it has to unwind, open. So who will do that work? Unwinding of the helix. Yes, helicase enzyme. It will be done by helicase enzyme. Now, once the DNA helix is opening, the bonds which are present that start breaking. Yes. So that breaking of the bonds, no, breaking of hydrogen bond will be done by DNA gyrase or topoisomerase. Done. Two things we got to know. For the replication process to take place, number one, the helix has to open. Unwinding should be done. Then breaking of the bond should be done. So unwinding work is done by helicase enzyme, and breaking of bonds will be done by DNA gyrase or topoisomerase enzyme will be doing that. So here 5 dash 3 dash 3 dash 5 dash. Yes. So whatever the information that is present here, no, whatever the information that is present here, if the DNA want to make the new stand, daughter stand, yes, for making the daughter stand, the primer has to be made. RNA primer has to be made. So that work of making the RNA primer, making the RNA, this is the short RNA primer. Information has to be copied. No, to copy the information we want RNA. Two strands together we are calling it as DNA. If we remove one strand, only one polynucleotide strand is present, that is RNA. No. So here one strand is having the information. So here this is the parental. No. Parental strand. Yes. So this is your template strand. No, real. That is coming from the parental strand. So information has to be copied. That should be copied in the RNA only. So when this short fragment is made, that is called as RNA primer. So to make this RNA primer, primase enzyme will support. Which enzyme? Primase enzyme. With the support of primase enzyme, first the RNA primer will be made. Is that clear? And once the RNA primer, if the RNA primer is continued long, no? So here, if you see one side I have connected, one side it is short fragments only. No? If it is continuous, we will tell continuous strand. No? Discontinuous strand. Or we can use the word leading strand. Here we will use the word lagging strand. Done? Yes. So the strand is ready. Whatever the information that has to be replicated, it has to be copied. No. So here synthesization should be taking place. It has to be synthesized. From where we are starting, for example, we need to start with the 5 dash. We need to start with 5 dash n. So assume this is the point from where the information starts copying. No, synthesis has to take place. So that point is called as ori site, origin of replication. Origin of replication. Yes. To copy the information, we will call it as polymerization process. When the information is copying from the parental strand to the daughter strand, no, it is called as polymerization phase. So in this polymerization process, what is happening? Whatever the sequence that is present, no, the nucleotide bases which are present, that has to be copied. Is that clear? So to copy, to polymerize the sequence, we have an enzyme called DNA polymerase enzyme. DNA polymerase enzyme. So here three types of DNA polymerase enzyme are present: DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3. Three enzymes are present. DNA polymerase 1 and 2, no, they are called as repairing enzymes. They are called as repairing enzymes to fill the gaps, to change with any of the nucleotide bases, to add or to remove. That repairing work will be done by DNA polymerase 1 and polymerase 2. And DNA polymerase 3 is mainly responsible for polymerization. Polymerization of the DNA sequence. Is that clear? Yes. So here the information, whatever the information that is present, that will be coded. Is that clear? It will be taken into the another strand. So once the strand, assume here the information is present, the information will be copied. No. So when the information is copied, from which strand it is getting copied? I told you this as template strand. Yes? No? So from to which it is copying? It is called as coding strand. It will be called as coding strand. Where it is getting coded, that strand will be called as coding strand. So once the information that is present here, it will be copied here. No? So now one side continuous strand I have showed. Another side we have short. No? When the fragments of RNA primer are short, no, these fragments are called Okazaki fragments. They are called Okazaki fragments. So these Okazaki fragments are joined by the enzyme called ligase. It will be joined and the full strand will be synthesized. So here completely if you cut, this will become a new DNA. From here this will become a new DNA. So from one parental DNA, two newly synthesized DNAs are produced. Yes, this process is called as DNA replication. It is called replication. It is done with the support of enzymes. Okay, whatever the enzymes I have written, any enzyme can be asked in the multiple choice question or in the fill in the blanks in your theory exam. Is that clear? Number one, helicase enzyme. Number two, DNA gyrase or topoisomerase enzyme. Number three, primase enzyme. Number four, polymerase enzyme. Number five, ligase enzyme. Totally six enzymes very important for supporting the DNA replication. Is that clear? Did you understand? Any doubts you have? No? It is clear? No doubt. Shall we proceed? Yes. So if you see the structure, it is appearing like a inverted Y, opposite Y. Yes. Y is like this. Yes. What I have drawn is opposite. So here this structure, Y-shaped structure, which we are able to see when replication is happening in the DNA. Once the unwinding of the DNA is taking place and when the replication process is taking place, the structure which is formed is called replicating fork. It is called replicating fork. In your textbooks, the diagram is given like this. Yes? Yes. So here I have shown you how the diagram will be formed. Once unwinding starts, the structure of Y-shaped structure will be formed. That structure is called replicating fork. Why? Because in that structure, replication is taking place. Therefore, it is called replicating fork. Yes. So, after studying about DNA replication, no, next process we need to study is very important process. Transcription. Done? Yes. Any idea? What is this transcription? What is transcription? Each and every cell, no, is undergoing this process in your body. Whenever you are eating food, no, without this process and without the next process, protein synthesis will not take place in your body. Okay? Each and every eukaryotic cell, whether it is a fungal cell, whether it is a prokaryotic cell, protista, whether it is a fungi, whether it is a plantae, whether it is animalia, all the cells are undergoing with this process. Transcription. What is this transcription? Hmm? Tell. No idea? No? This chapter is not done in your colleges? Yes, it is there for your midterm exams. Nay, it is done in your college? Yes, tell. Something you tell. What you have understand? You have not understood? Huh? You have not understood? क्या बीच नहीं समझ में आया सर? हाँ? नींद आए थे कि? ये बहुत अच्छा चैप्टर जी है. 12 to 17 marks from this one chapter you can score. ठीक है? एंड तुम्हें समझिए तो नींद नहीं आती. अब रेप्लिकेटिंग 4 कराया ना, नींद आई. हाँ? अब ट्रांसक्रिप्शन जब करातून तुम नींद नहीं आती, खुश होती. और ये क्या सर कर बोल. हाँ? उसे पूछ रहा हूँ क्या कर को, समझ में आया नहीं आया बोल को. नहीं, डोंट नो. यस यस. सो इन अब हमें क्या प्रोड्यूस करे. सो इन रेप्लिकेशन व्हाट वी हैव डन. रेप्लिकेशन में क्या करें हमें. एक डीएनए से और दो डीएनए को बनाए. नहीं, फ्रॉम वन डीएनए वी हैव प्रोड्यूस टू मोर डीएनए. उसे हमें क्या बोल रही. रेप्लिकेशन. नहीं, अब इन ट्रांसक्रिप्शन, in short, keep it in your mind, transcription means RNA synthesis. We are producing RNA. We are producing RNA. So how we are producing RNA? In this process, the information which is present, the information which is present in the DNA strand. This is your DNA strand. Yes, this is your DNA strand. Nee. So whatever the information that is present, assume A, T, G, C, A, T, G, C. Nee. So this strand will act as template strand. This strand will act as template strand. This strand will act as coding strand. It will act as coding strand. So here, what will be the basis here? Opposite. Now, A will be T, A, C, G. T, A, C, G. Yes, we have complete DNA strand now. In the transcription process, from this DNA helix, DNA strand, we are producing RNA. We want to produce RNA. RNA synthesis is done. What we are doing, whatever the information that is present in the template strand, no, that will be copied in the mRNA. It will be copied in mRNA. Nee, I told you in the last class, 3 types of RNA are present. Do you remember? Yes, mRNA, mRNA, tRNA, and rRNA. mRNA is messenger RNA. tRNA is transfer RNA, and rRNA is ribosomal RNA. ठीक है. So whether you are producing, whether you are synthesizing, whether you are preparing tRNA or mRNA or rRNA, that process is called transcription. ठीक है. So what we will do, whatever the information that is present, no, that will be recorded in the mRNA. It will be recorded in mRNA. What is there here? Yes. So what we will copy? We will copy T. Sure. Yes. Yes. RNA में T आता है. In DNA, thymine is present. In RNA, so what will be coded here? 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 Yes. Yes. The strand which you have made, no? The information that is present in the DNA strand, no? That is copied to the mRNA. The process is called transcription. Okay? The information that is present in the one strand, parental strand of the DNA is copied to mRNA. When it is copied to mRNA, mRNA is synthesized. No. So in short, we will tell it is RNA synthesis. Did you understand? समझ में आया? Yes. To undergo this process, अब ये transcription process करना बोले तो we want a transcriptional unit. We want transcriptional unit. I have taken this DNA strand. Yes, I have taken this DNA strand. To this DNA strand, to run the process of transcription, we want a unit. The transcriptional unit is nothing but the tools or device required. Is that clear? So, transcriptional unit consists of number one, very important topic. Okay, either 2 marks they can ask you give the schematic representation of transcriptional unit, or they can ask you explain transcription process in prokaryotes or transcription process in eukaryotes. Okay, yes. So number one, promoter. Number two, structural genes. Number three, terminator. So the transcription unit consists of 3 parts: promoter, structural gene. Yes, we have taken a DNA strand, complete DNA strand I have taken. This is the template strand, this is the coding strand. Yes. Now assume I want to start the process from here. I want to start the process from here. To start the process, promoter is required. To start the process, promoter is required. From where it is starting? This is your promoter. It will be promoter. And where it is ending? It is ending here. Yes, terminator. It will be terminator. So for this, remember always, whenever the transcription process is taking place, it will start from the 5' end upstream. 5' upstream. Okay. So the process will start from 5', 3', and when it is ending, 3' end is downstream. Ending. No? So the process will start in the 5' polarity, 3 polarity. Is it clear? When it is ending, the stand will be in the 3', 5'. You are able to see the process is starting from here. So 5', 3'. No? Ending 3', 5'. Yes. Where it is starting? Promoter. Where the process is ending? Terminator. No? And if you see one more, I have written structural gene. What is the structural gene? Any idea? Yes. Did I write any where there structural gene? No? Is there any gene there structural? Which genes are called structural genes? Yes. There are so many genes. No, in our body, if you take, there are so many genes involved in different functioning. Yes or no? So depending on the function, we will classify. Yes, there are few genes which are involved in mutation process. No, if the genes are involved in mutation process, what we will call mutant. Yes, if they are involved in replication process, we will call them as replicon. Multiple choice question will be asked. If it is involved in recombination, yes, yes, recon. Yes. If the genes are involved in functioning, yes, the genes are involved in functioning, no, they are cistron. These cistrons are nothing but the structural, the genes which are involved in function, functional genes, the genes which has to be coded, required genes from where the information has to be copied, that genes are structural genes. Is that clear? The structure which I have made, no, with the promoter, terminator, template, coding, structural gene, this whole structure is called as the schematic representation for transcriptional unit. Did you understand? Transcriptional unit. And how many genes we discussed now? Yes. The genes involved in mutation is, replication is, recombination is, functional is cistron. Yes. Now, if you take any doubts still here, everything is clear. Now, to understand the process, two processes we have to study in transcription. One happening in bacteria, one happening in eukaryotes. Yes. So, now functional genes I have written as functional genes. Functional genes. So here, when we study about these functional genes, we are discussing about cistrons. Which gene? Cistron. And these cistrons are different in prokaryotes and eukaryotes. Yes, what is the difference? Any idea? Yes? No? In the prokaryotes, these cistron genes are polycistronic. Polycistronic. They are polycistronic. In eukaryotes, they are monocistronic. Yes. Now, coming to what is the difference between this polycistronic and monocistronic, sir? No? So here, if you take the particular stand, yes, listen here. If you take a particular DNA segment, no? So here, assume continuously the genes are same. The genes are same. Means the whole segment has to be polymerized. There is no change in the segment continuously. We will classify this cistron genes into two forms. It will be classified into two forms. For the genes which has to be coded, we will call them as exon. Which will not be coded, which is not involved in coding, no, we will call it as intron. Yes, जो जीन्स कोड होना उनको एक्सॉन बोलते हैं, जो नहीं कोड होना यूज नहीं है उनका इंट्रॉन्स. In the bacteria, no, continuously we don't have introns, only exons are present. नहीं, poly किसे बोलते सो? Many. Poly का तो many. Continuously many exons are present. No introns are found in between. Therefore, polycistronic genes. किसमें? Prokaryotes. Multiple choice question will be asked here. And coming to the eukaryotes. Eukaryotes, if you see here, one segment of exon will be present, one segment of intron will be present, one segment of exon will be present, one will be intron. So here one, therefore we will tell monocystonic. Done? Did you understand? Yes. Shall we proceed with the process now? Yes. So here transcription process in prokaryotes. Prokaryotes बोले तो we are talking about bacteria. Yes. So to start with the process, what we want? Yes. We want one strand of the DNA. Yes. We want one strand. Nay? So what we are doing? We will take two. Shall I draw the straight line or shall I draw helix? Helix draw करो ऐसे straight draw करो ऐसा संदे नहीं helix करो आपको नहीं. Yes. So we have taken a complete DNA helix 3 dash 5 dash 5 dash 3 dash. Yes. Complete DNA helix we have taken. Helix. Yes. To undergo with this transcription process, we want a promoter, we want a terminator, we want a structural gene. So all the three are ready. Yes. So where it has to be copied? It has to be copied in the cistron. Nahin? So we are taking RNA polymerase enzyme. We will take RNA polymerase enzyme and sigma factor. Yes, we have taken DNA helix, promoter is ready, terminator is ready, structural genes are ready. Along with that, we are taking RNA polymerase enzyme, sigma factor, and we want mRNA. Done? Yes? Now, shall we start with the process? Yes? When the process begins, that is the first step now. So, first step, we will call it as initiation. Initiation. Yes. To start with the initiation, where it will start? So, sequence should be in the form of palindrome sequence. Mirror image. Data box कहती हमें. TA कैसा पढ़े तो भी? Same. नहीं. Data box or palindrome sequence. Assume from here the process is starting. To start the process, we want sigma factor 1. Yes, to start the process, sigma factor is required. This sigma factor will start from where it is starting. That point is the promoter. Yes, RNA polymerase enzyme is copying the information. It is copying the information from the template strand to mRNA. Yes. Once the process starts, it is initiation. First step is initiation. To start with the initiation, we want sigma factor, we want RNA polymerase enzyme and a promoter. Done? Yes. Now, segment by segment it is happening. Now, it is in the mid segment. Assume there are 10 helix. We have reached 5. We have completed 5. We are in the middle. No, center. That means that will be your second phase. That is elongation. It is elongation. Done? Finally, we have reached the end phase. No? That is terminator. To terminate the process, to start the process, we took sigma factor. Nay, to end the process, we have RHO factor, rho factor. Yes. So once the mRNA started, there was a plain mRNA where no information was present. Only the primer was present. RNA primer was present. Now that is completed into a fully polymerized, fully sequenced mRNA. Is that clear? So after termination, what will happen? DNA helix is separated. DNA helix is separated. The RHO factor is separated. Yes, RHO factor is separated. The mRNA strand, fully polymerized strand is separated, and RNA primase polymerase enzyme is also separated. Done. So this last step is called as termination. So the process of copying the information from one strand of the DNA parental strand to the mRNA, this whole process is called transcription. Done? Did you understand? Yes? Easy, Henny? So three processes are involved, that is initiation, elongation and termination. Done? In the initiation, the process starts. In the elongation, the process is moving on, adding up of the nucleotides. Is that clear? So finally, coming to an end, it is termination. To start the process, sigma factor. To end the process, RHO factor. Done? To run this process, RNA polymerase enzyme. Done? Any doubts? समझ में आया? नींद आई? नहीं आई? नहीं? Easy, बहुत easy है. समझ गया तो there is nothing in this. ठीक है? Yes. Shall we go with the eukaryotes now? Yes. The transcription process in prokaryotes was so easy because the mRNA was having which form of genes? Yes, polycystronic genes. It was having polycystronic genes. But when the formation of the mRNA takes place in the eukaryotes, directly mRNA will not be produced. It will not be produced. Instead of mRNA, hnRNA will be produced. Shall we proceed? You don't know chapter? Huh? This is fifth chapter. Molecular basis of inheritance. Hmm. You are in second year PUC. Hmm. Uh, this is fifth chapter. Molecular basis of inheritance. Last class we started, and we are in the center of the chapter, middle of the chapter. Yes. Done. Yes. Now coming to the transcription process in eukaryotes. Yes. So, as I told you, in the eukaryotes, we have monocystronic genes. Monocystronic genes. That means when the mRNA is synthesized, once the process starts, no, initiation, elongation, termination takes place, no, the mRNA which we have to get, but we are not getting mRNA, we are getting hnRNA. Is it clear? Heterogeneous RNA. Is that clear? Short segments of the RNA, that is called heterogeneous RNA. So here, if you see, the diagram is given like this in your books. No? Yes? So, proper diagram is not given. Complete initiation, elongation, termination is not given. No. So here if you see, the elongation phase is shown where the information is getting copied into the mRNA. So once we get this mRNA, when we take this mRNA is not mRNA, but this is HNRNA. This is HNRNA. After the process of transcription, initiation is over, elongation is over, and termination is over. After the complete process of transcription in the eukaryotes, what is the result we have got? HNRNA. Okay. So why we are calling HNRNA? Because in this RNA we are able to see both. Assume this is the exon, this is intron, exon, intron, exon, intron. Yes, both the segments are present. Exon is the coding segment, intron non coding. Yes, non coding. No. So here when we talk about the process first, initiation हुआ, elongation हुआ, termination हुआ. In the prokaryotes, that process when it started, some factor was responsible. Yes, sigma factor was responsible to end the process. But in the eukaryotes, no sigma is present, no RHO is present. No sigma, no RHO. Okay? Who is doing this work, sir? RNA polymerase itself will complete the process. It will start by its own, it will end by its own. Okay? This RNA polymerase enzymes are of three types. In eukaryotes, they are of three types. RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. Done? Yes. So here, listen carefully. RNA polymerase 1, if it is involved, if in eukaryotes, if RNA polymerase 1 is involved, it is responsible to produce tRNA. It is responsible to produce tRNA. If RNA polymerase 2 is involved, responsible to produce mRNA. If 3 is involved, responsible to produce rRNA. This apart from this rRNA, some srRNA and a small ribosomal RNA. We will call it as small ribosomal RNA and snRNA, small nuclear RNA. Done? Did you understand till here? Any doubts? Which RNA is involved? That RNA will be formed. Which RNA polymerase is involved in the process? That if polymerase 1 is involved, what is the end product we will get here? tRNA. 2 is involved. If 3 is involved, done? Did you understand? Yeah. So I'll stop here. We'll continue after namas. Done? Yes. Shall we continue? Yes. So we were discussing about transcription process in eukaryotes. So once the process is started, there is no sigma factor to start the process. Initiation happens by its own, and then elongation will take place, and finally termination will happen by its own. So finally, the RNA which is produced is hnRNA, heterogeneous RNA, which are the small segments of mRNA, not fully complete mRNA, but it is the segment of mRNA. So what should be done now to convert this hnRNA into fully matured mRNA? No, so additional three steps are involved in this process. To complete the process and to make mRNA out of this process, three additional steps are involved. Number one, capping. Number two, splicing. And number three, tailing. Three steps are involved. So here the hnRNA which we have 5' end, 3' end, hnRNA. So in this hnRNA we have exons, we have introns. Yes. So the first step which we will take, the first step is splicing. It is splicing. So we will take an enzyme called spliceosome. Spliceosome. So this enzyme will go and bind with this, which one? Exon, intron. It will bind with intron. Yes. So it will bind with this intron and it will remove the intron. So that process of removing the introns, no, that process of removing the intron from the hnRNA is called as splicing. Yes, first work we have done. So once we remove all the introns from the hnRNA, the size of the hnRNA will be same. It will become still shorter. No. So here we have got this. No. So what we have to do? We have to add the nucleotide bases. Right? So what we will do, from one side, from the 5' end, we start adding methyl guanosine triphosphate. We started adding the methyl guanosine triphosphate. Is it clear? So one by one we are adding. From which side? From the 5' end. And again what we will do, from the 3' end, no, we start adding adenylate residues. So 170 to 200 adenylate. This adenylate residue is nothing but adenosine monophosphate. Another name for adenylate residue, adenosine monophosphate. So here from the 5' end, when we are adding the methyl guanosine triphosphate, no, the process is called capping. It is called capping. From here what we are doing is capping. Yes. From the 3' end, when we started adding the adenosine monophosphate, no, so here the structure is increasing, no. So when it is increased, that means tail is increased. This is the head part, that is the tail part. So formation of tail is formed, no. So we can use the word poly a tail is formed. Is that clear? Poly, a tail is formed. So here we can call the process as tailing. Tailing process will take place, lead to the formation of poly, a tail. What is added? We are adding adenylate residue. That means we are adding more number of adenosine. No, so here polyadenosine monophosphate residue. Okay. So from two sides, complete addition work is done, capping is done, tailing is done, splicing is done. So finally, once the capping process and tailing process is completed, the mRNA is the hnRNA now is fully transcribed mature mRNA. Done? Did you understand? Yes. So the whole process of making RNA is called as transcription. In prokaryotes, it is simpler because they have only one form of structural genes. In eukaryotes, it is quite complicated because directly mRNA is not produced, hnRNA is produced. Again, we need to make the mRNA. So additional 3 steps are involved. That is splicing, capping, tailing. First step is splicing. Second is capping. Third is tailing. In capping, what we will do? We will add methyl guanosine triphosphate. In tailing, what we will do? We will add adenylate residue, that is polyadenosine monophosphate. This process is called capping and that process is called tailing. And here splicing, when do we call splicing? When the spliceosome is involved and it is removing the intron, the process is called splicing. So after these 3 steps, the mRNA which is formed is the fully transcribed matured mRNA. Done? This whole process is called transcription process in eukaryotes. Done? Any doubts? अब समझ में आया? नींद आई? नहीं नहीं? Easy होगा नहीं? हाँ, दो प्रोसेस, दो में से एक 5 मार्क्स को आते रहता है. Either they can ask you explain the process of transcription in eukaryotes or prokaryotes. Prokaryotes very easy. May eukaryotes you need to explain that first, then the other additional 3 steps. Done? Yes. Yes. Now we know DNA replication. Yes. We know transcription. Yes. So when the DNA replication was done, right? Who proposed this DNA replication process? Watson and Crick. When he explained the double helix model, he proposed DNA replication and he called it as DNA as semi-conservative mode of replication. Okay. To prove that, yes, when the DNA replication takes place, it is in the semi-conservative form. No? So, 2 more scientists by name Messelson and Stahl. No? They experimentally proved. That DNA is semi-conservative in nature. What do you understand by this word semi-conservative? Yes. Yes. So here you have studied principle of inheritance and variation. Yes. When we took 2 plants, one tall plant, one dwarf plant, the result was it's a hybrid, intermediate. No. So here same thing. No, when the replication takes place, according to Messelson and Stahl, no, the DNA is semi-conservative in nature. Why we took one parental strand, right? One DNA strand, and with that one DNA strand we got two new daughter DNA strands. Nahin? So in that two new daughter DNA strand, one is parental, one is newly synthesized. Nahin? It is not same. It is 50-50. No, conservative means fully parental. Semi-conservative, it's an intermediate half. To explain this, Messelson and Stahl experiment is called Messelson and Stahl experiment. Is that clear? Semi-conservative mode of DNA replication experiment, both are same. If they ask you explain semi-conservative mode of DNA replication, no, it was proposed by Watson and Crick, but explained by Messelson and Stahl. So in this experiment, what they have done, very easy experiment, right? Just they wanted to see what is the result when the generation to generation continues in the formation of DNA. Is that clear? So they have taken E. coli bacteria. Which bacteria? E. coli bacteria. Okay? And this E. coli bacteria was grown in the medium NH4Cl medium, ammonium chloride. So in this ammonium chloride, they have taken the heavy isotope of nitrogen. This is heavy isotope of nitrogen. Is that clear? Now, when they started growing this E. coli bacteria in the medium, first generation took place. Multiplication of the bacteria. When the ammonium chloride is having nitrogen atom with the heavy isotope 15, no, when the multiplication of the bacteria takes place, that will be continued. Yes or no? It will be continued. So when the DNA after the first generation, no, we were able to see both the strands of the DNA is having n to the power 15, n to the power 15. So what Messelson and Stahl did, they have taken one more NH4Cl medium. They have taken one more medium of NH4Cl, and in this they have taken the light isotope, that is normal isotope of nitrogen, that is n to the power 14. They have grown the bacteria. So in one plate, in one culture, the bacteria is grown in the n to the power 15 medium. In one they have grown n to the power 14 medium. Now, if a conservative mode of replication is seen, that means all the strands should have either 15 or 14, either 15 or 14. No, but these two were mixed up. Both the medium was mixed. Now n to the power 15 and n to the power 14 is together. Is it clear? When the DNA replication takes place, no, after 20 minutes, first generation, this was first generation, 20 minutes. Okay. What is the result we are able to see? So here it was 50-50. The DNA which is produced, no, is having one strand of n to the power 15, one n to the power 14, n to the power 15, n to the power 14. No, the result what we have got is intermediate. That means what has happened? When the replication took place, one is parental strand, one is newly synthesized strand, one is parental strand, one is, this is one strand, this is one more strand. Nay. So when n to the power 15 is also present, 14 is also present, when replication takes place, half will be 15, half will be 14, half will be 14. Yes. So again to check the result, right, one more generation was made, one more time. So how did we do this, sir? So during after the replication is over, we have used cesium chloride, CSCl medium. ठीक है, that is cesium chloride to separate this DNA segments. Is it clear? So from that we got this first generation result as intermediate result. Now assume if you take this intermediate result and again undergo with one more generation, what will be the result now? Yes, we have taken 2. Now what will be the result? 4 we will get back. Yes, all will be the intermediate now. All will be the intermediate. All will. I have taken one DNA of n to the power 15 medium. One is 14. 14 is nitrogen, 14 atom is present. That is normal isotope of nitrogen. Is it clear? And 15 is heavy isotope. Is it clear? Now, if both are present together, now, what is the result we got? 50-50. 50-50. So, intermediate or hybrid, we have got the result. Now, again, if I cross this intermediate for 40 minutes, what will the result I get? Yes, n to the power 15, n to the power 14, n to the power 14, n to the power 14. Yes, n to the power 14, n to the power 14, n to the power 15, n to the power 14. Yes, what is the result I got? 2 parental strands, again 2 hybrid. Why? Why? Any idea? N to the power 15 is, I told you, it is the heavy isotope. Separately, if it is present, it will be continued. Same result we will get. If it is present together, it will not be splitted. It will be present together. It will be expressed together. Separately, it will not be expressing itself. No. So here, n to the power 15, 2. In that we are having intermediate, and 14, which is a normal isotope. Nay. So they have separated. Is that clear? Now assume one more cross for 80 minutes. If you do, what will be the result? Yes, 8 strands will take place. Nay. 8 will be there. 8 will be there. Nay. Yes. In that, how many will be n to the power 14 separate? N to the power 15 will not be separate. N to the power 15 will not get separate. It will be in the hybrid form only. So how many n to the 14 we will get, and how many intermediate 15, 14 together we will get? Yes. Yes. It will be 2, 15, 14 reduction will take place, and 6 will be 14, 14 only. Did you understand? Yes. In the NEET exam, once this question is been asked, okay, what will be the intermediate in the 80 minutes of time? Is that clear? Done? Did you understand? So here with this we got the confirmation that when replication takes place, semi-conservative mode of replication takes place. Okay? The next strand which is produced, it is not conservative in nature, it is semi-conservative in nature. Did you understand? Any doubts still here? Yes. So in this chapter, 4 very important topics we have studied. Number 1, Griffith experiment, transforming principle. Number 2, Hershey and Chase experiment. Number 3, DNA replication, Watson and Crick model. And number 4, transcription process. Done? Yes. So when we study DNA replication, and when we study transcription, no? Both the topics we know now. Yes. Will you differentiate what is the difference between these two now? 2 mark question. They can ask you differentiate between DNA replication and transcription. Yes. We know we have studied DNA replication also. We know what is transcription also. Nee? How will you differentiate? What is happening in DNA replication? Yes. How many parental strands are involved? Yes. One parental strand? Yes. So here, one parental DNA, not strand. One parental DNA giving rise to two daughter DNA molecules. Done. Here, one strand of DNA produces mRNA. Produces mRNA. Done? Yes. How many polynucleotide strands are present here? Here, two polynucleotide strands are involved. Here, only one polynucleotide strands are involved. Which bases are present in DNA replication? Which bases are involved? A, T, G, C. Here, A, U, G, C. Which enzyme is involved? Yes. Which enzyme? Here, DNA polymerase enzyme is involved. Here, which enzyme is involved? RNA polymerase enzyme. Yes. Where DNA replication is taking place? Yes. Where it is taking place? Where? In the nucleus. Yes. In the nucleus. Only nucleus. Yes. And it can take place in the cytoplasm also. Here, transcription process is taking place in the only nucleus. Done? Did you understand? If they ask you for 2 marks, differentiate between DNA replication and transcription, at least 4 points you have to write. Done? Any doubts you have till here? Any doubts? No doubts? Yes? Yes, yes. Prokaryotes. Because a well-defined nucleus is not present in prokaryotes, it can take place in the cytoplasm, fluid that is present. Nay, nucleoid is present, therefore, the replication will take place in the cytoplasm. Done? Yes, we know DNA replication, we know transcription. Yes, shall we solve some questions? Yes. Find out the correct match from the following. Find out find out the correct match from the following. Column 1. A. Helicase. B. Gyrase. C. Primase. D. DNA polymerase 3. Column 2. Number 1. Joining of nucleotides. Joining of nucleotides. Number 2. Opening of DNA. Number 3. Unwinding of DNA. Number 4. RNA priming. Option number 1. A2, B3, C4, D1. Option number 2. A1, B2, C3, D4. Option number 3. A4, B3, C1, D2. Option number 4. A2, B4, C3, D1. Option A. Helicase enzyme. Opening up DNA. Gyrase. Unwinding of DNA. Primase. RNA priming. And DNA polymerase. Joining of nucleotides. The strands of DNA which is synthesized continuously during replication is called A. Lagging strand, B. Discontinuous strand, C. Coding strand, D. None of the above are correct. None of the above are correct. Answer is? What will be the answer? No, I am asking answer. What is the stand called? Leading stand. DNA polymerase enzyme was discovered by A. Arvind Khurana. Arvind is an Indian. Arvind Khurana. B. Niranbarch, Scottish name. Niranbarch. Nirenberg. Niranbarch. C. Watson and Crick. D. Cornberg. Korn. Corn. Berg. Berg. Cornberg. Yes, what is the answer? It is C. Watson and Crick. What they have done? Yes. They propose what? Double helix model. Okay. Semi-conservative mode of replication. What is the question asked? Yes. The RNA polymerase enzyme who discovered? DNA polymerase enzyme who discovered? Option? Arvind Khurana and Niranberg. They have proposed coding system. Triplet coding system, meaning genetic code. These two have proposed that. This Kornberg is a scientist who has proposed, who has discovered DNA polymerase enzyme. Yes, experimental system used in the studies on the discovery of replication of DNA has been number one, Drosophila melanogaster. Drosophila melanogaster. Number two, Streptococcus pneumonia. Number three, Neurospora. Number four, none of the above are correct. None of the above are correct. Yes. So which organism is used in DNA replication? We are using Drosophila melanogaster. What experiment is conducted on Drosophila melanogaster? Who took? Yes, very good. Chromosomal theory of inheritance was experimentally proven by T.H. Morgan by using Drosophila melanogaster. Then, on the Streptococcus pneumoniae bacteria, which experiment was conducted? Repeat experiment was conducted. Transforming principle experiment. Neurospora. No? There is one hypothesis called 1G, 1 enzyme. 1G, 1 enzyme hypothesis. So on that, they have used Neurospora species. Okay. To conduct replication of DNA, which bacteria? It is E. coli bacteria. So option D is the answer. None of the above are correct. So the correct answer is Escherichia coli. It's called E. full form Escherichia coli. A DNA molecule, a DNA molecule makes complete turn, makes complete turn after every A 20 angstrom units. Option A 20 angstrom units. B 3.4 angstrom units. C 10 bases. D 12 bases. Yes. So option C 10 bases. So once a complete turn is taken, it will have 10 bases. The diploid content of human DNA, the diploid content of human DNA is, it is a repeated question in multiple choice in theory. They can ask you haploid content or diploid content. The diploid content of human DNA is A 3.3 into 10 to the power 9 base pairs, B 6.6 into 10 to the power 9 base pairs, C 4.6 into 10 to the power 6 base pair, D 5386 nucleotides. 5386. 5386 nucleotides. Yes. Option B. Option A. Option C. Option D. Yes. So option C 4.6 into 10 to the power 9 6 base pair, that is for equal length. And 3000, what I told you, 5826, that is for flex 174 bacteriophage virus. Okay. So 2 I have written option 1 and option 2. Option 1 3.3 into the 10 to the power 9 is haploid content and 6.6 is diploid content. Which was the first genetic material evolved? Number 1 RNA, 2 DNA, 3 protein, 4 both 1 and 2. It is RNA. How many questions? Sigma factor, sigma factor is the component of is the component of A. DNA ligase, B. DNA polymerase, C. RNA polymerase, D. All the above are correct. Option C. RNA polymerase. Functional unit of gene. Functional unit of gene that specifies synthesis of one polypeptide. Synthesis of one polypeptide is number one, recon, number two, muton, number three, replicon, number four, cistron. What is wrong about transcription in eukaryotes? What is wrong about transcription in eukaryotes? Number one, transcription is monocistronic. Transcription is monocistronic. Number two, hnRNA undergoes additional process. hnRNA undergoes additional process called capping and tailing. Number three, one RNA polymerase complete the process. Number four, initiation factor and termination factors are associated. Option 4 is the option 4. 4 is the correct answer. There is no initiation factor or no termination factor. Option 3, RNA polymerase, which I told you. Okay. They are 3 RNA polymerase, but if it is tRNA, 1 will be involved. If it is mRNA, 2 will be involved. And if it is snRNA or srRNA, it is 3 involved. है ना, दो दिया था ना. तो additional process है तो बोल रहे हैं. Yes. While working on neurospora, while working on neurospora, Beadle and Tatum, scientists, Beadle and Tatum proved that number one, plant cells are totipotent. Number two, DNA replication is semi-conservative. Number three, every gene is responsible for specific enzyme. Every gene is responsible for specific enzyme. Number four, virus also have genetic material. Option 3. So the hypothesis proposed by Beadle and Tatum is one gene, one enzyme hypothesis. According to them, each enzyme is controlled by particular gene. So option 3 is the correct answer. That's right. You stop here.